Dear colleagues, this is the nervous system again at the cellular level, and I'm having to repeat this in order to make a shorter video. So I'm going to break this into two parts. And we're here looking at the neuroglia, and both actually the central and peripheral nervous system. And you'll see me get to both of these. And here we are now. These are the cells that create the tissues of the central nervous system, which is the brain and the spinal cord, other than the neurons. Okay, so here we have the astrocyte, which is actually, you can see, holding both capillaries and cell bodies of neurons, as well as the white fiber processes, the dendrites and the axons, in appropriate positions so that the nutrients get to the cell bodies of the neurons, and waste products are also reabsorbed. Okay, so this is basically the scaffolding and architectural feature of the nervous system. Then we have this next cell down here is the microglia, and this is actually a macrophage. It's a monocyte that has diapedized or exited the circulatory system. And what we have here is the macrophage with its dendrites. These dendritic cells are particularly active. And so this is a very active macrophage, and it's cleaning up cellular debris and any sort of infection that might pass through this way. One of the details that we'll learn about in the circulatory system is that the blood travels from arterial to venous through the skull, actually from the nasal cavity straight through the meninges. There's channels that we'll observe, venous channels, sinuses actually in the meninges. And so it's good to have these macrophages there as phagocytes. Then we have the ependymal cells. These are interesting because they line the fluid-filled ventricles, and they have these little hair-like devices called cilia that have automatic beating in a particular direction. They're quite organized, and they cause flow to go from the lateral ventricles to the third ventricle to the fourth ventricle to the subarachnoid space. And these cells are also responsible for creating the cerebrospinal fluid. That is to say, they are transformed in certain areas into capillaries, where the transudation of fluid from blood to CSF occurs. And so these are very interesting cells. I'll come back to that in a moment when we look at CSF. Here we have a different kind of scaffolding. This is actually a cell called an oligodendrocyte. You can see here's the nucleus and the cytoplasm, and then there's these extensive outreaching sort of devices, pseudopods as it were, that wrap around, and we actually create sort of an electrical tape around the white fiber processes of the neurons. And this actually protects the signal, and it also increases the speed of the signal. Very important. The oligodendrocyte is unique to the central nervous system. We have something else that does this in the peripheral nervous system. Down here we have both satellite cells and we have Schwann cells. The satellite cells, as you can see, are surrounding this cell body, and we're out in the peripheral nervous system where the cell bodies collectively arrive, or that is to say they're occupying something called a ganglion. So here we have the dorsal root ganglion, for example, in a sensory cell. Here is the information coming in, and then it's proceeding on inside the spinal cord. So coming from the outside and going into the spinal cord. These cells right here are called Schwann cells, and they're actually protecting the white fiber dendrite or axon. So those are the options here, and we refer to that as the fiber, the nerve fiber, right? And so these are collectively creating a kind of an electrical tape, but you'll notice that there are bare spots, and we'll get to that in just a second as we turn the page here. And we take a look at this large neuron here with Schwann cells attached to the axon. Okay, so this particular cell body would be in the central nervous system in the spinal cord, most likely. In some cases, it will be in the brain, but the ventral root of the spinal cord is where we find many of these, and they're motor neurons. In this particular case, these are all dendrites. These are incoming, or that is to say, they direct incoming signals, action potentials. And then there's one outgoing, only one outgoing, and that would be right here in this portion of the cell body. You'll notice it's clear of contents. There's contents throughout the cell body. Besides the nucleus and the nucleolus, there's these things called Nissl bodies, which are actually, if we take a look here, rough endoplasmic reticulum. What we know about that is that we're producing proteins, in some cases enzymes that will take an active role in the creation of neurotransmitters. So to some extent we're actually producing neurotransmitters. In some cases we're making enzymes that play a role in the biosynthesis of neurotransmitters. Okay, the neurotransmitters will collect here in the hillock. You'll notice there's no Nissl bodies here, no rough ER. And then, by the way, we used the term Nissl bodies before there were electron microscopes because we couldn't see that these things were rough ER. That's why there's this earlier nomenclature. Anyways, the neurotransmitters are going to actually flow down these little microtubules, little channels, all the way through the axon, and they will collect at the axon terminal down here, where they will arrive just at the tips here of the axon terminal where there's going to be synapses. And when it's time for the signal to cross over the synapse, the neurotransmitters will effect that particular outcome of crossing the synapse. So that's a slight delay. It's fast when it's an electrical event, and it's slow when it's a chemical event. Okay, and notice that we have Schwann cells here every so often, and the Schwann cells are leaving little spaces, and what happens is if we have an action potential, an actual signal develop here, there's a crossing of the membrane that I'll get into momentarily of ions, sodium and potassium ions, creating an electrical event, an EMF, and the electromotive force is just large enough in order to be able to depolarize over here at this next nodal point. Okay, so it just so happens that that's the way it works. The EMF is just large enough to cross this distance, and it depolarizes here. Then this EMF depolarizes here, and then this EMF depolarizes here. This speeds up the channel, or that is to say the action potential. It goes from 1 meter per second if there were no Schwann cells here to 100 meters per second if there are Schwann cells here. So that's quite an improvement in speed. Okay, so you can understand why, for instance, muscular sclerosis could be a problem if we're, how would I say, engaged in autoimmune destruction of these things, because that will change the speed, and therefore our habits and reflexes will be completely different and altered. Meanwhile, this action here, the impulse, is referred to as saltatory conduction. Okay, across, that's the increased speed event. The nodes here are often referred to as the nodes of Ranvier. Okay, and we're going to conclude right here in order to make this a small enough unit to go on YouTube.